As Tomasz said, I'm working on red blood cell damage. But if you don't know what that is, it's not a problem. I will introduce the problem to you and maybe also try to show you why this is an interesting problem and why more work needs to be done in this field. So this is the general agenda. First, I will, as I said, give you an introduction. If you don't know about red blood cell damage, why this is an issue, then I will show you how we attempt to model this, because there are a lot of different stages involved in the modeling. Then I will show you two example simulations where we try to predict the blood damage and then compare it to experiments. Then I have two, let's say, more in-depth topics, which are on the one hand, uncertainty quantification, and on the other hand, rheology, which is the work that I've been doing with Tomasz. Yes, and then I will summarize the topics. So let's start with the background. Blood, if you don't know, consists of many different components. The red blood cells are the largest by volume. So there's the blood plasma, which is the actual fluid. And then 50% or 40% of the total blood volume is made up of these red blood cells, which are in their initial resting shape, biconcave discs, as you can see sketched on this slide. And they contain hemoglobin within. So there's some viscoelastic membrane around the hemoglobin. Then within the cell is this fluid, which is called hemoglobin. Now, generally, this hemoglobin is contained within the cell, and so it should not be present in the free bloodstream. So it's an issue once this hemoglobin is released from the cell into the bloodstream, because then on the one hand, the cells cannot transport the oxygen as well anymore. So this impacts their function as red blood cells. And on the other hand, this hemoglobin in the blood is also toxic to various organs, so to the kidneys, for example. So you don't want this hemoglobin in the free blood. And this is what happens during red blood cell damage. So how does this occur? Generally, at rest, when there's no blood flow, the red blood cells tend to aggregate, and these structures are known as rouleaux. So in this state, a bunch of red blood cells can be stacked on top of each other. Under shear, these rouleaux tend to break up, and in this state, the cells can move through the flow somewhat more independently, and this is called tumbling, because the cells can tumble in this motion. At even higher shear rates, this tumbling stops, and eventually the cells become oriented with respect to the flow direction. And this state is called tank treading, because in this state, the cells are assuming a fixed orientation, and the membrane of the cell is rotating around the contents, or around this hemoglobin, which is contained within. And this is kind of like the motion of the treads of a tank. And that's why it's termed tank treading. And eventually, at even higher shear rates, this tank treading continues. But the higher the shear rates, the more elongated the cells become. And eventually this hemolysis starts to occur. So then the membrane is strained so much that pores can start to form in the membrane. And then the hemoglobin, this fluid which is contained within the cell, can leak out through those pores. So this is really the state of hemolysis, which we don't want. In general physiological circulation, shear rates are relatively low in the fluid. So this is not an issue generally. But in any type of blood handling medical devices, which introduces more artificial types of flow, with higher shear rates, this can be an issue. So when people get, for example, these blood pumps, which are implanted to the human heart, they tend to expose the blood to much more shear than what's usual in the human circulation. And this is where we need to account for this phenomenon, and where we need to try to predict the occurrence of this hemolysis in advance based on CFD simulations, in order to limit the occurrence of this. Now, why do we need to work on this? At the moment, this is still really hard to predict on the computer. So there is one example, which is this FDA benchmark blood pump. So this is some example geometry that was designed by the FDA in the US, and it was handed out, and basically various laboratories were tasked with predicting the hemolysis in this pump without knowing the results before. And generally for the velocity field of the blood, so the CFD simulations were relatively accurate. You can see here that the various participants had errors of up to 10%, which is, I guess, not perfect, but it's at least in the range of what's realistic. However, the hemolysis, which is the second diagram, was widely off between participants. You can see here that participant 3S has an error of 10,000%. some other errors were 100%. And so there was a big variance between the predictions, and none of them were really accurate because there is essentially no determined way at the moment, or no established way to predict this accurately. And this is why we want to work on this hemolysis modeling to improve such predictions. Now, how does this work in general? We always need 3 steps for this hemolysis modeling. The first step is always CFD, which essentially gives you a flow field for the blood in the domain. So for example, in this blood pump, we would first do a CFD simulation to obtain the velocity gradients. Even in this CFD, you can have different choices. I will, for now, just focus on incompressible Navier-Stokes, but you can also think about more complex rheological models here. And the goal of this is to get the velocity gradients or the shear forces within the fluid. Then those go into the so-called red blood cell model. So here you need to somehow assume some behavior of how the red blood cells will respond to these forces, to these stresses in the fluid. And there I will present to you 3 different approaches. We will come to these in a minute. And the goal of this is always to predict the so-called effective shear rate or effective shear stress, which is then not the fluid shear rate or fluid shear stress, but takes into account the response of the red blood cells to these stresses. And from this, we can then apply a so-called hemoglobin release model. So the hemoglobin, which is this fluid that's contained within the cell, is released through the cell, and essentially this depends on the deformations of the cell. So once we have this red blood cell model, which tells us how deformed the cells are in response to these stresses, then we apply this model, which gives us, depending on the deformation, the release of hemoglobin into the blood. And in this stage, we will also need more experimental data because this phenomenon is very hard to capture exactly numerically. Okay, so let's go through these 3 stages that I outlined here: CFD, red blood cell model, and hemoglobin release model. First, CFD. As I said, for now, I will just focus on the simplest case, which is Newtonian incompressible Navier-Stokes equations. In this context here, I also want to highlight this rate of strain tensor E, which will play a role later, which is basically just the symmetric part of the velocity gradient. We use an in-house stabilized finite element code to simulate this. And we stabilize this using the variational multiscale method, which also gives us a way to incorporate turbulence, basically. For rotating machinery, such as these blood pumps, we use the multiple reference frame approach. So this basically allows us to do a steady simulation and to account for the rotational forces in a rotating frame of reference without actually having to move the mesh. Okay, so this is now just the basis, and this talk is not so much about CFD, so I will go over this relatively quickly for now. Let's say we successfully simulated the blood flow, and we now want to simulate how the red blood cells respond to these flow forces. And so far, this is just a one-way coupling. So we assume the CFD solution is given by this Newtonian rheology, and then we want to model the red blood cell behavior in this flow field. So the simplest approach is to simply use the so-called Bluetooth stress. So in this case, we have the 3-dimensional stress states, so these stress tensors at each point in the domain, and we just reduce them to a simple scalar, which we call this effective stress here, by applying some sort of norm on this rate of strain tensor. And in this case, this norm looks like this. So it basically places some weight on all the different components. This is similar to the von Mises stress in solid mechanics. So it's just a way to reduce this tensor to a scalar. And this assumes that the material of the red blood cells is completely elastic, because essentially, as soon as you have some stress that applies to it, this effective stress will immediately reflect this, because it's just a norm on this rate of strain tensor. So this assumes an instantaneous response to any fluid stress. On the other hand, we have so-called strain-based models. And for this, I will start with the simplest form, the Kelvin-Voigt viscoelastic model, where you don't have just an elastic response, but a viscoelastic response. In this case, this is no longer a scalar equation, an algebraic equation, as we had before for the norm, but now this is a differential equation. And you can see that now this effective stress, sigma effective, can vary with time, and also you have to solve an ODE to compute it. And it depends on the instantaneous fluid stress and the current effective stress. And this instantaneous fluid stress we again compute using this Bluetooth norm as in the previous example. So this is what I call the simplest form of a strain-based model. And these are called strain-based models because here we try to predict actually the membrane strain of the cells rather than just looking at the fluid stress. So the other models are called so-called stress-based models. And we here have this characteristic deformation time tau, which governs how fast the cells respond to changes in the fluid stress. So one example, simple shear flow, so that's the Couette flow essentially in 2D. Now, if we apply these models to it, we basically assume in the beginning the cells are not deformed at all. We now, with different values for this characteristic deformation time, you basically control how quickly the cells obtain this deformation that's imposed by this velocity gradient. So with small values, tau equal to 0.05, this response is very fast. With larger values, this response becomes slower. And with this Bluetooth model that I showed you on the slide before, the response is essentially instantaneous. So this tau goes to 0 because it's a purely elastic response. So this is the first slightly more complex model. To visualize the difference, also in a maybe more applied case, again, Bluetooth is the elastic response, Kelvin-Voigt is the more viscoelastic response, and we call this stress-based versus strain-based. We now look at an example blood pump. So on the right-hand side, you can see some geometry for a blood pump with an impeller, which will now rotate, and you will see the trajectory of a single red blood cell as it travels through this domain. You can see it travels towards this impeller, and then it gets moved towards the outlet. And on the left-hand side in this diagram, you can see how or what stresses the cell experiences. So the blue line here are the Bluetooth stresses, and you can see that these peaks are very sharp because the fluid stress varies rapidly along this trajectory. And on the other hand, you can see if we apply this Kelvin-Voigt model, this viscoelastic model, along this trajectory, then the response is somewhat more slow. And also you can see that the 3 different peaks have different effects. So the third peak in particular, this peak here, has the biggest or induces the biggest deformation of the red blood cell compared to the others because the cell is already deformed to begin with here. So in this sense, this viscoelastic model introduces some sort of memory, some sort of history of the strain, which the elastic Bluetooth model doesn't have. So we can also have accumulation of strain over several cycles. Now, why is this important to account for these viscoelastic effects? For this, we've done experiments with the Charité University Hospital in Berlin. And they basically have some small, they call them microchannels, but it's more centimeter scale. And we compared 2 different geometries here. One reference channel, which is just a plain channel, and one channel with a sudden constriction in the middle. And you can see here that the sudden constriction channel induces high shear stresses within this constriction because the fluid velocities are high, and so there's large gradients of the profile there. So if you apply a stress-based model, which only looks at the fluid stress, then you would predict that there's high hemolysis in this channel because of these high gradients here. On the other hand, if you apply a strain-based model, you get much lower values. Basically, if you look at the scales here, this is up to 500, this is up to 10. So these values here are much lower in this contraction. The reason is that essentially the cells move through this contraction so quickly that they don't have sufficient time to respond. As I showed you before, this characteristic deformation time for red blood cells is usually on the order of 0.2 seconds. So it takes up to 0.2 seconds to really react to changes in the stress. And the cells move through this constriction within milliseconds. So this is why through this viscoelastic response, you get much lower cell deformation. And actually, if we now do these experiments, these were just the simulations. We did experiments and we compared the relative index of hemolysis. So how much more hemolysis occurs in this channel compared to this reference channel. And essentially, the stress-based model predicts much higher hemolysis by a factor of 100. The strain-based model predicts almost no additional hemolysis because this exposure time here is so small. And if we run these experiments, we really see that there is almost no hemolysis in these constriction channels, despite these high stresses. So this is why it's important to not just consider the fluid stress, but also consider the history of the stress and the model the viscoelastic response of these cells. Okay, and then there's a class of even more detailed red blood cell models, which we call morphology models, where we actually try to model the shape of the cells. So in this case, we use some mathematical analogy. We assume that every cell is ellipsoidal. Now, as I showed you in the beginning, this is not always completely true. So these cells at rest are generally biconcave disks, which is not an ellipsoid. But the assumption is that these states don't really matter, right? We want to predict the hemolysis, which occurs when the cells are highly stretched. In this highly stretched states, they resemble ellipsoids. So if we now assume that cells are always an ellipsoid, every ellipsoid can be described by a tensor, which we call the morphology tensor S, which is symmetric, positive, definite. And the eigenvectors of this ellipsoid, of this tensor, represent the orientation of the ellipsoid, so the three major axes. And the eigenvalues represent the lengths of these axes. So a larger eigenvalue means more deformation in the direction of this axis. So just by this 3x3 SPD tensor, we can describe the shape of this ellipsoid. And the model, this Aurora morphology model, then looks like this. So it's a transport equation now, or in this case, just an ordinary differential equation, because we essentially apply this along a trajectory of a single cell for this morphology tensor, which looks like this. And you have different effects here. On the left-hand side, this was called Yarman derivative. And this essentially represents the frame of reference that's moving along with the cell and also rotating along with the cell. So this omega tensor here depends on the eigenvectors of the cell. So this V is the eigenvectors of this tensor. And you have to take the material derivative to obtain the rotation. Then on the right-hand side, you have all the effects of the fluid. So on the one hand, the first term is the shape recovery term, which will relax the cell back to its initial shape in the absence of any stress. The second term determines the influence of the rate of strain tensor, which aligns the cell with the principal axis of strain and stretches the cell along those axes. And then the third term is the effect of the fluid vorticity, which rotates the cell. And so you have some linear superposition of all of these effects. And then there is one more model, which I call the tank treading model, which is essentially a multi-scale approximation of this more complex model. I don't really want to go into details here, but this model is the same kind of effects as the original model in a more efficient framework. So I will just compare the two models in this example of a Couette flow here. So in this Couette flow, we have a fixed lower wall and a moving upper wall, and we introduce red blood cells here at the inflow, and we then follow them along this upper wall and see how they deform as they move along this upper wall. Here's an example red blood cell, and we now move along with the cell in this frame of, so we basically see the cell always at the center, so the frame is moving with the cell. And what happens is basically instantaneously, you can see very quickly the cell aligns to some state. So this initial state was just chosen arbitrarily as we introduced this. So this could be, for example, from a previous flow that the cell is coming from. And essentially what happens quickly is the cell aligns to some orientation. And this is the tank treading orientation that I showed you before. So this fixed orientation in the flow that the cell is transported at. This happens almost immediately, and then over a much longer time scale, the cell starts to deform to basically respond to the magnitude of the shear rate in this flow. So you can see that there are these two different scales, the orientation scale or the alignment scale, and then the deformation scale. And this, because of this discrepancy in scales, this Aurora model is very hard to simulate numerically because this alignment scale is so short and the deformation scale is so long. So really you need time steps in the order of magnitude of this alignment scale to always capture the rotation and alignment of the cell accurately. but you still need to simulate for as long as the deformation scale, so you get the correct deformations, which are what drives hemolysis in the end. So this is why simulating both at the same time becomes really challenging. So what we do instead with this tank treading model, TTM, is we basically just assume that the cell is always aligned. So we can compute this aligned orientation, this tank treading orientation, with some algorithm. Again, I don't want to go into details, but this basically skips this initial alignment step and allows us to use time steps on the order of magnitude of this deformation.
I have a question.
Yes.
So concerning the position of the cell in the channel,
yeah.
the center of the cell is fixed, or the cell can move up and down in principle?
In principle, it can move up and down, but in this quet flow, the streamlines are horizontal, right? So the cell
but still you can have lift.
Yes, this is a very idealized case. Okay. So in this case, it's just plain shear flow. But yes, in reality, cells tend to move towards the center of the channel.
Yeah, that's what I was thinking.
Okay. So this really helps us reduce the time here by 1 order of magnitude, because, as I said, we can now choose larger time steps to simulate this. And you can also see here that the results on the long time scale of the deformation are essentially the same. So we don't really introduce any error through this method. Let's compare now these more complex strain-based models, where we try to actually model the shape and orientation of the cell, to the more simple Kelvin-Voigt model that I showed you before, which is just a scalar, linear, viscoelastic ODE. What you see in simple shear, and this is again this delayed deformation with some time scale, is that, again, both models are essentially the same. So in both cases, we have this response to some stress impulse with some delay, which is governed by the internal time scale. So here, this doesn't make a difference. The main difference is if you have extensional flow. So this occurs, for example, in hyperbolic contractions, where the stress now applies along the same direction as the flow. In the quet flow that we had before, the flow is this way, and the stress essentially occurs in a 45-degree angle to this direction. But in this extensional flow, the stress occurs in the same direction as the flow. And this makes a difference, because cells then are aligned in the same direction as the stress. So this stretches them more. And there are, in fact, experiments which also show that in such hyperbolic contractions, the same stresses lead to more deformation and to more red blood cell damage. And this is where the difference occurs. So here, this tank treading model predicts the same, the Kelvin-Voigt model predicts the same deformation in the asymptotic scale as the Dirkes model, which means we only look at the fluid stress to determine how much the cell is deformed after sufficient time. Whereas the tank treading model here predicts higher deformation because, as I said, the cell is now aligned with the axis of stress. So this is an important effect that we can only integrate with these more complex models, which also model the orientation of the cell. So we capture the difference between shear and extensional flow. Now I have some graphic to illustrate this hierarchy of models. The most simple model was this Bluetooth stress, which takes into account the fluid stress, but nothing else. And we call this stress-based model. Then we have the strain-based models. First, the Kelvin-Voigt model, which has this additional parameter, the characteristic deformation time, which governs how quickly cells respond to changes in stress. And then the more complex models, these morphology models, where we try to actually model the shape and orientation of the cell. This was the Aurora and TTM model. And here we have this additional effect of the orientation, which allows us to distinguish between shear and extensional stresses. So in the next parts of the talk, I will mostly be referring to these two classes as stress-based and strain-based models, because these two models often lead to similar predictions in practice. So we really now just want to compare these two. Okay, so now we have this deformation of the cells based on these red blood cell models, and we now want to determine how much hemoglobin is released from the cell depending on their current deformation. For this, there are two approaches. The most commonly used one in literature is called the power law model. So this is really just a simple law. This IH is called the index of hemolysis, and this is just a measure for how much hemoglobin is released from the cells. And in the simple form, we have three parameters, A, alpha, and beta, which are just fit to empirical data. So essentially, these are done using some rheometers. So it's like an outer cylinder and an inner cylinder, and you rotate some cylinder at a certain rate, which introduces some shear in this gap. You know the shear rate exactly because you know the rotation rate of the cylinder, and you expose the cells for a certain amount of time. So you also know for how long you expose them. And so with this, you control this sigma and the T, and then you just measure the hemolysis in the lab. And then from this, you get these sort of curves. So you get a lot of points, and then you try to fit a curve through this 3D space to basically fit the points as well as possible. And this is how these parameters are determined in these simple experiments. And then the same parameters are used to predict hemolysis in more complex geometries. One of the issues is that in this form, this is only valid for constant stresses over this exposure time t, because this is just an algebraic equation. Once you want to apply this to real cases, the stress will always vary, because in some red blood cells, the cells are not constant, and the cells that the stress that a single cell experiences highly varies over time. We saw this blue curve before, which had these strong peaks and rapid changes in the stress. So really, this is not constant over this exposure time t. So what people usually do is some linearization in time. So they essentially define this linearized index of hemolysis by taking the regular index of hemolysis to the power of 1 over beta. What happens then is this power of the t is just 1, and the 1 over beta is on this. And then what they do is they take a time derivative, assume that this stress is constant over this small time, and then basically this t drops out and only this term remains. This is, however, not true in general, because as I said before, this sigma effective can vary rapidly, so it's not constant over any interval in time. So really, this is a bad approximation, but this is the only way to apply this power law to realistic geometries. And for a long time, this was the only model that was available, because nobody really can simulate all the details going on at the microscopic scale. So really, this was what was used. But as I said, there are some issues mathematically with this law. So instead, what I want to propose is the so-called pore formation model. This is a slightly more detailed microscopic model. So here you can see that this is already in differential form, so we don't get this issue anymore with trying to convert from this algebraic form to a differential form. We don't need to linearize anything. We again have two free parameters here, this h and k, and we also fit this to data with the same experiments. What we have here in addition is this AP, which represents the total pore area. So with this, we actually try to predict how pores form in the membrane at certain deformations. And then there's this function, which is constructed using some biochemical considerations, which basically determines how many pores you have overall in the membrane at this deformation state. And there's also some threshold. So you can see here before this deformation, there are no pores at all, and then it starts. So you can integrate more microscale effects into this model. And this is also valid for non-constant stresses. So the hierarchy here is the power law is really purely empirical. And then there's this pore formation model, which has some physiological basis or some microscopic considerations. So far, all these models I presented have been in the Lagrangian frame. So basically, we've looked at single cells, and we've tried to apply these models along these trajectories of single cells. And this is why we always had these ordinary differential equations here for the path lines, or here for the effective stress, or also for the hemolysis index. If you do this, then you need to usually track cells through the domain and then compute this index of hemolysis by averaging over all of the cells. What we tend to do instead is use a so-called Eulerian approach. So we simply use the velocity field from some CFD simulation, and then all of these equations are not ODEs anymore, but they become PDEs. They become advection equations, essentially. So we now don't want to determine these values for each single cell, but we want to determine them in each computational mesh element. And then this represents sort of an average over all the cells that flow through this element. And as I said, then these become PDEs. To visualize this, we can look again at a simple blood pump. So the Lagrangian approach here would consist of tracking all of these cells at the inlet, following them through the blood pump, and then evaluating this model along each trajectory. So after sufficient time, you would have something like this. And here the colors indicate the deformations of the cell that we have along these trajectories. The issue with this approach is that even if you have a lot of path lines, a lot of cells that you follow, there will always be some gaps in the coverage. And in fact, there can be regions of recirculation which you don't penetrate at all because you pick the cells at the inlet. So with this Eulerian approach that we use instead, we get a field for this deformation, which looks like this. So we get a value at each point inside the domain, even in these recirculation zones. And this makes it easier to then use this to optimize the design of these pumps because we really see the whole domain. So how do we do this in this Eulerian frame? As I said, these are PDEs and not ODEs, so we can't just use a simple ODE solver. So what we do is these are essentially tensor transport equations. So we aggregate all of the tensor entries, which are up to 6, because in this Aurora model, we have this symmetric positive definite 3x3 tensor. So these are 6 independent components. And we aggregate them all into one vector phi, which contains these components. And then this is essentially an advection equation for this vector phi. And the source term f here then couples the different entries of this vector to each other. We have some Dirichlet condition at the inlet, and we have Neumann conditions on the walls and on the outlet. And we solve this using some Galerkin formulations. So we have stabilized finite elements as for the CFD. So this is here, these are the Galerkin terms with the residual. These are the stabilization terms. As I said, we use VMS stabilization here. And then this is the so-called discontinuity capturing term. And this is used to capture sharp gradients in the solution, which can occur, for example, close to the inlet when you have high inflow. And then next to the inlet, you can have some recirculation with cells which are already deformed. And the inlet introduces fresh undeformed cells. So between this, you can have sharp gradients. And what happens if you don't do this is overshoots and undershoots. So you might get unphysical values with extremely high or negative deformation along this jump. And so this discontinuity capturing term smoothes out these gradients in these sharp zones. Now I have 2 examples where we apply these 3 steps that I showed you before. The first one is the FDA pump. This is the example I showed you in the beginning where the FDA designed this pump and the hemolysis predictions were widely off by all the participants. So the first step is CFD using incompressible Navier-Stokes. This is the flow field here, and this is not from me. This is from a previous paper, which was already validated using PIV data. So I don't really want to go into detail here. Second step is now to apply this red blood cell model on top of the flow solution. And you get here 2 different fields. This is the stress-based model. And you can see that here there are some sharp gradients, whereas the strain-based model, this Kelvin-Voigt model, leads to a much more uniform or much smoother distribution. And this is because of this characteristic deformation time, which basically always smoothes out these stress peaks and leads to the cells slowly responding to changes in stress. So in this sense, the strain-based model smoothes out these stress peaks. If we now apply the hemoglobin release models to this pump, so to the red blood cell deformation, we have 6 operating conditions here, which the FDA defined. And these gray bars are the experimentally measured values at these 6 operating conditions. You can see that the standard approach in literature, which is the stress-based red blood cell model combined with the power law for the hemoglobin release, gives really bad predictions. So again, here we are off by a factor of more than 100 for all of these operating conditions. And this is what essentially most people use at the moment. If we use a strain-based red blood cell model instead with the same power law, we're a bit better, but not so much better. And now if we use this strain-based red blood cell model together with the pore formation model, so with this more microscopic scale hemoglobin release model, then you can see that we're actually within the same order of magnitude and even mostly within the standard deviation of the experiments here. So this is one of the first times that actually we can predict hemolysis in the absolute sense, not in some relative variation between operating conditions sense, in advance with computational methods. Another example is the FDA nozzle. So this is some geometry that was also defined by the FDA. You can see the cross section here. So fluid flows in here, then there's a sudden contraction, and then it widens again. And we also simulate this. Here you can see the mesh, and here you can see the validation of the CFD solution. And what happens is you can see that the flow becomes very turbulent inside this contraction right after this sudden contraction. And if we now apply these red blood cell models to this, you can see that in the stress-based model at the top, there are a lot of critical areas, a lot of high shear rates. So these are the red zones. But in the strain-based model at the bottom, in this Kelvin-Voigt model, there are really only two critical zones. There's this zone here right after the contraction. There's some recirculation zone, and there's a similar zone at the outlet. And what happens in these zones is essentially this accumulation of strain that I was talking about earlier. So because of this history of stress that the strain-based models introduce, here the strain can accumulate because red blood cells tend to recirculate and tend to be exposed to these high shear rates over and over again. And so this is why this region is critical. And similar, the region at the outlet here is also critical because again, there's recirculation and these high shear stresses apply over and over again. So this is how these strain-based models can also help us find critical areas in the flow which aren't so apparent if we only look at the fluid stress. So again, we then apply these hemoglobin release models, and you can see these are the experiments. This is the best result I found in literature, which is also using a special form of the stress-based power law, but here they used special coefficients which were tuned to similar flow geometries. But you can see that these are still relatively far off from the experiments. In fact, this is a little bit better than the standard approach from literature, which is the stress-based power law. But you can see that only the strain-based pore model is again within one standard deviation of the experiments. And this is really important because we use the same coefficients here as for the FDA pump before. And these coefficients were not tuned to these specific experiments. They were taken from these simple shear chamber experiments that I explained before. So in a way, this can be universally applied. So you can fit the coefficients only once based on these experiments, and then you can apply this to many different geometries. So this is why this is an important result. Now I want to talk about two more applications of these models. First, uncertainty quantification. So if we go back to the hemoglobin release models and specifically the power law, I told you how we usually obtain these parameters A, alpha, beta from these experiments, basically with this curve fit. The issue is that there are many different sets of these parameters A, alpha, beta. So many different laboratories have done the same experiments, but they always get different values for these parameters. So there's a lot of variance in the data and also in the parameters that are used to fit the data. And essentially, we even saw that sometimes if you take out one data point, the value of these parameters can change quite a lot. So these are very sensitive to the data because this model is essentially just a very simple representation and doesn't capture the underlying physics. So usually what is done, we have this experimental data. And what you do usually is you do this deterministic curve fitting, which then gives you one value for each of the parameters. So this gives you one set of parameters. And then from this, you can do these simulations, which is also what I did in the previous section. And then you get one value for this predicted index of hemolysis. This is the standard approach. What we do in this uncertainty quantification approach is we try to get distributions for the probabilities of these parameters. So we don't just want one set of parameters, but we want to see how likely is it that we have this value, how likely is it that we have this value, and so on. And then from this, you can also get a probability distribution for the predictions from your simulations instead of just one single value. So how do we do this? This is the mathematical problem formulation. So we have some model M, which is in this case the power law model, which depends on these parameters theta and independent variables X, and we have data Y. And we now want to find the probability that we have the true parameters theta, given that we have these observations Y. And this is just the Bayes theorem that I wrote down here. So essentially what you need to compute is you need to get this probability that we have these parameters if we don't have any prior evidence, and this is called the priors, times the probability that we see these observations given these are the true parameters, which is called the likelihood function. And here in the denominator, you have the total evidence, which is basically the probability, not assuming anything, that we see these observations. And this is the hardest to compute. And in fact, this cannot be computed analytically. So what we do here is we do some sort of numerical approximation, which is called Markov chain Monte Carlo or MCMC. I don't want to go into details, but basically what this gives us is it gives us samples of this distribution. So instead of analytically determining this distribution, we can sample it from it. So we get a bunch of parameters which are sampled according to this probability. And what we get from this is curves like this. So these are the numerically determined parameter distributions. You can see now for each of these three parameters, this is A, this is alpha, this is beta, we get these curves, and essentially this mean value of the curve is similar to what we would get in the deterministic setting where we have only one value for each parameter. But now we have these curves. What's interesting here is also this correlation plot. So you can see that A and alpha are strongly correlated. And this is one reason for why we see so many different parameter sets from different groups, because essentially any of these combinations along this line work well. So you can have a slightly higher value of alpha and a slightly higher value of A, and it fits the data almost as well as a lower value of both. So you can basically choose many different combinations along this line. Okay, so this is now the inverse uncertainty quantification, where we have data and we have experiments, and we want to compute the parameter distributions of the model. This is called inverse UQ. What we do then is so-called forward UQ. So we now want to propagate these distributions through the simulation. So essentially we have this flow field for the blood pump, and now we take these samples. As I said before, with this MCMC method, we get many samples from these distributions. We get many different sets of parameters. And in this forward UQ, we basically just have to evaluate this simulation for all of these different samples. And this then gives us the distribution of the results. And in this case, this is very easy because this index of homolysis is computed just as a volume integral over the solution. So we can compute this as a post-processing step, and all the different parameter sets are independent of each other. So we can simply calculate all of these volume integrals in parallel. And so we evaluate this typically for about 100,000 samples, 100,000 volume integrals. But as I said, on a big cluster, this is easily doable because it can be parallelized. What we get first for this FDA nozzle is these kind of distributions. You can see that these two forms of the power law both have very wide probability distributions. And this basically shows that depending on which parameter set you use, depending on your data, you might get values somewhere between 10 to the 4 and 10 to the minus 1. So that's a huge range in the possible predictions that this power law can give you. On the other hand, these Poor-based models have much narrower ranges. And you can see, in fact, also again, that this train-based Poor model overlaps with the experimental standard deviation. So this is, again, this shows how uncertain the power law is, and one of the issues with just fitting these parameters from the power law to experiments. And finally, the same for the FDA pump. These are, again, the 6 operating conditions, and you can again see that the blue and orange, so these two power laws, have wide ranges, whereas the Poor model has much narrower range. And again, this strain-based Poor model, which is red, overlaps with the model uncertainty or with the experimental uncertainty almost everywhere. Okay, and then finally, I want to come to rheology. So far, I've only ever used this incompressible Newtonian Navier-Stokes equations for all these results. A slightly more advanced step would be to use the shear thinning model, where the viscosity depends on the shear rate, which is also something that's observed in blood typically, mostly because of these rho lows that we see, which start to break up, and this then reduces the viscosity of the blood. But really, I want to go one step further and see how we can couple the rheology of the red blood cells to the fluid flow. And for this, we focused on this Rajagopal viscoelastic model, which was formulated specifically for blood more than 20 years ago. And essentially, this consists, like the Navier-Stokes equations, of this momentum equation. Then there's this extra stress equation, which contains the pressure, the viscous parts, and then this part, which is the so-called stretch tensor B, which represents the stretch of some fluid element. And for this B, we have a separate transport equation, which looks like this. And if we now compare this to the Aurora model, which I showed you before, which was this morphology model where we approximate red blood cells as an ellipsoid, it looked like this, if you remember. And you can see that this looks pretty similar. So now instead of this BE, we have this S, which is just a different nomenclature. You can see that the first term, this recovery term, is essentially identical. The second term, which governs the response to stress, is almost identical, except for this coefficient F2. The third term, which governs the response to vorticity, is almost identical, except for this F3, and also that we have to here subtract the rotating frame of reference from the vorticity. And we have this additional term, which is related to the frame of reference rotating along with the cells rather than along with the fluid vorticity. So these are the two differences, but the models do look very similar. So the idea here, what we try to show is that these models can in fact be written in a unified form. So we then decomposed the tensors, so this S for this and the B for this, into their eigenvalues and eigenvectors, and then rewrote the equations in terms of these eigenvalues and eigenvectors. We get a new set of equations. And we, in fact, show that both of these models fit into this framework with only 4 different coefficients. And in fact, this C2 and C3 and C4, these coefficients are commonly the same. So they only differ in these 2 coefficients for the deformation equation. And this is work that we've done with Tomasz Bodnar, and we have a paper for this under review. And in fact, if you then compare these 2 models in this corrugated channel, you can see that both models highlight similar regions in the flow as critical. So you can see that the absolute values here that we obtain for the deformation are still much different between the 2 models, which is just due to these different coefficients. But the areas in the flow which are regarded as critical are at least qualitatively similar. So then the hope is that in the future, one can find such critical regions for hemolysis directly from this viscoelastic solution rather than having to run this separate red blood cell model on top. And this would make it much easier for people who are already working with viscoelastic flow solvers, because they wouldn't have to get into these red blood cell models. The solution could be inferred by the flow solution immediately. So this is the final model hierarchy then for CFD models. We have these incompressible Navier-Stokes equations, which only do fluid momentum transport and also mass conservation. Then there's the shear thinning model, which takes into account the shear thinning nature of blood through this viscosity. And then the more complex viscoelastic model, which also can have shear thinning coefficients, and in addition to this can also account for viscoelasticity in the fluid. And so basically it takes into account the microscopic elastic response or viscoelastic response of the red blood cells. Okay, so to come to an end and to sum up the different parts, I first talked about CFD, and CFD is always the basis for all of these models because the flow field is the driving force of hemolysis, and the flow field really governs how the red blood cells deform. And in the end now, I also showed you how we can do the inverse direction. So how the red blood cell behavior also can affect the flow field through viscoelastic models. And then I showed you different choices for this red blood cell deformation model, in particular strain-based models, where we actually try to model the behavior of the red blood cells and the area strain of the cell. And we try to model orientation as well for complex cases where the shear stress and the extensional stress are not the same. And then I showed you different choices for the hemoglobin release model, where especially this pore formation model is much better than the current standard, which is the power law. And it yielded accurate predictions for the absolute values of hemolysis in different cases, even though it was tuned on very simple experiments. And also with the uncertainty quantification, we saw that the uncertainty in this model is much smaller than in the more commonly used power law models. Yeah, and with this, these are the references. I want to thank my sponsors, and I want to thank you for your attention.